Good afternoon, everybody. My name is Philippe LAMBIN. I'm a radiation oncologist, and I'm chairing this very exciting session. You will have two short talks on new technologies that you probably don't know. Right. So I would like to start with telling you a little bit about a sort of quite a new discipline, technology, which is born about less than 5 years ago, which is called radiomics, and which we believe could help for drug development. So we all know that tumors are very heterogeneous, and we have to recognize that if all precision medicine is therefore based on one single biopsy, we could have a problem, especially we are interested, for example, in a tumor microenvironment, because usually those biopsies are taken randomly in a very heterogeneous tumors and not necessarily representative of the whole tumor heterogeneity. So it has been, by the way, shown in the group of Toronto that if you want to have a good picture of what's going on for hypoxia, acidose, and other microenvironment endpoint, you would need at least 5 biopsies. So I'm sure that our lung specialists would not be happy with if I request systematically 5 biopsies. So we believe that the solution is actually imaging because it's non-invasive, it's 3-dimensional, and you can also repeat it because we all know that tumors evolve differently with the stress of a treatment. So there are different ways to innovate with imaging. The first one, very expensive to buy new hardware. If you can convince your management to spend $5 million for MR PET. You can also spend several years to develop new biomarkers, for example, HX4 to look at hypoxia. But we believe that there is also another very interesting way, which consists to use new software to try to extract more information from the imaging we already have. So the first radiomic hypothesis is that we are actually able to extract quantitative information, because you will agree with me that when you read the radiology reports, we are usually very happy when there are a few dimensions quantified in the report, and the rest is very qualitative. So here the idea is that we can extract more information, quantify them, automate the process, and also extract information which are not visible by the eyes. And to illustrate this concept, I will show you a short animation of 2.5 minutes.
In cancer, we face at least 2 challenges. Every tumor is different, and there's an ever-increasing number of treatment options available. To personalize treatments optimally, we need to be able to identify the biological differences between tumors. Biomarkers provide just such a solution. The goal will be to find these biomarkers in a way that's easy to perform, non-invasive, low cost, and captures the 3D complexity of solid tumors. We believe radiomics delivers all of this. Radiomics is the analysis of standard clinical images from CT, MRI, or PET scans using original software to extract more than meets the eye. It uncovers hidden biological information from standard scans. We've been collaborating with teams in the Netherlands and the US to look at lung, head, and neck tumors. By putting their size, shape, and texture measurements through complex software, we were able to map out each lung tumor's unique features. We discovered that a subset of the tumors shared 4 characteristics related to intra-tumor heterogeneity, and these cancers grew and proliferated faster than others, leading to worse patient outcomes. What's more, the exact same 4 characteristics, the same signature if you like, showed up in aggressive head and neck cancers too, again predicting a poorer survival rate after radiation therapy. Our now patented and published findings mean that at very little cost, standard medical scans can reveal more about tumor behavior than traditional clinical methods for tumor staging, just by processing the scans through radiomics software. And there are petabytes of scans sitting in the imaging databases of hospitals waiting to be mined. Our vision is for all this patient information to be fed into an ever-growing online database that doctors around the world could use to make better treatment decisions.
So just to illustrate the concept, and also to make clear that we also try to capture information imaging you don't see, I just show here these two squares, and if I ask you which square is the darkest, you will say what an obvious question, but actually they are exactly the same darkness, it's just an illusion. So our point, and we demonstrate that as well, is that this situation occurs regularly as well in imaging. So how does it work? Basically, the first step is to segment the region of interest. It can be a tumor, but it also can be a normal tissue. Actually, for normal tissue, it's easier. Then you extract about several thousand of features, which are related to intensity, shape, and texture. Then you make an analysis, and you have a report. So the principle is to transform imaging, which at the moment mainly a qualitative type of research, into a sort of a mineable data set. And to extract information could be used for diagnostic, for predictions, and prognostic. So these are the features: intensity, shape, textures, and after processing the image with filtering, you can also extract more information. And just to give you some examples here, you have, for example, here two tumors, different shapes. Even so, the first one is probably smaller, but there is it's obviously linked to biological behaviors of these tumors. Here you have tumors with different intensity. You see at the above panel, you have a large spread of intensity, and in the under panel, you see a very narrow spread of intensity. That's also a quantitative representation of different heterogeneity of those tumors. And even so, the first one is probably smaller, it's much more heterogeneous, and that also tell us something about the biology of this tumor. You can also extract quantified texture. There are about 100 different ways to do it, and get some quantitative information. So 2 years ago, we published this paper. That was probably the first one where we really demonstrate, I think, that there was a real value in this approach. And the first experiment we did was really very simple. We just look at in the x-axis of different patients with lung cancer, and the y-axis we look at the radiomics features. And we just ask the computer to rank with an unsupervised clustering approach these different patients. And we were very surprised to see that actually most of the adenocarcinoma were in the right on the corner, and the squamous cell carcinoma in the left side of the graph. So that was for us the first evidence that in a simple CT without contrast, there is actually biological information. And later on, we are even now able to detect with a not 100% accuracy, obviously, but some, for example, EGFR mutations in this subgroup. So, of course, there is one big risk of radiomics, is that you have a sort of a overfitting risk. You always have something significant because you have a lot of features. So we were very worried of that, of course. So in the first experiment, what we did is try to use different data sets with test-retest data sets where patient requests to sit on the bench, then stand up, and then sit again with each time imaging. And also, we had data sets with delineation by different doctors, a big variation. And all the features which were not robust, reproducible across these different experiments were eliminated. So try to keep a sort of a pool of features which we trust, which we know they are robust. And we also identify some features which are very dependent of the vendors, which are different with Philips G, etc. So finally, we end up with a pool of robust features, and we took the top features, which are the most prognostic in the group of tumor intensity, tumor shape, tumor texture, and wavelet after a different filtering, and we generate a signature. Then we test these signatures on independent data set. And these four features are linked to energy, compactness, gray level, non-uniformity. It doesn't matter too much. What is important is that we capture with a sort of unsupervised approach features which are related to intratumor heterogeneity and irregularity of the tumor. And when we test that on a totally different data set coming from Nijmegen with a different CT from Philips, and we just rank on the mean, we could see a prognostic value. And there is a continuous variable, so you can actually identify a subgroup who does very badly, and even some stage 2, inoperable stage 2 for medical reasons, and some T4, for example, are doing very well. And some centers already use it now when we have this discussion with inoperable stage 3 non-metastatic lung cancer to see whether we have to go for heavy radiochemo or more for a palliative approach. And just for comparison, this is the predictive value of TNM that we use in our daily practice to make this decision, and it's absolutely no predictive value. That's known because TNM has been developed mainly by surgeons to look at operability and doesn't tell you a lot about outcome after radiochemo. So in other words, the message here is that a tumor which are heterogeneous are doing bad and also have a higher proliferation. I will come back on that. So then we ask the question, what now if we take exactly the same signatures and we apply blindly without any processing on a data set of head and neck cancer? Some of my colleagues were very upset that we even were considering to do that, saying that head and neck is a different disease, there are air cavities, etc. But we did it. It doesn't cost a lot of money to do this type of research. That's the advantage of radiomics. And actually it was working even better than with lung cancer. And that's not surprising because there are also some gene signatures, for example, the wound, proliferation, hypoxia, which is also working across different diseases. So to try to understand the biological base of this, we had another data set where we did operate the patient. We had high-quality CTs, gene expression array, and to make a long story short, we found that this radiomic signatures with only 4 features is actually related to tumor proliferation. So another radiomic hypothesis, which is probably more daring, is that not only we can quantify data, extract prognostic data, but we can also imputate what is going on at the genomic level with a simple CT of MR without contrast. And one proof of concept that we did is just to try to guess the HPV status of patients with head and neck cancer. And that's an unpublished work, but the accuracy is not very high, but it's highly significant and validated in 2 different data sets. So that could be useful in certain cases with metastatic patients. You cannot have biopsies of all the locations to see whether HPV status is still, phenotype is still as dominant as initially. But it's also, for me, a proof of concept that we can get some information from what's going on at the genomic level with a simple imaging, but a right software. What about North American? Why do I ask that? Because when I presented those data in the US, usually some colleagues told me, we don't believe you. I said, why? Because there are more than 1,000 patients, but no North American in it. So, okay, fair enough. So we were fortunate to have access to more than 600 patients, oropharynx treated in PMH Toronto. Again, different CT, different protocols. And the bad surprise is that half of them had dental artifacts. For some reason, the HPV plus patients go more often to the dentist, and half of them were HPV plus. So we just tried, and again, without any processing, any calibration, blindly, and again, it was working significantly, and again, continuous variable. So this just to tell you that we believe that this approach is very robust, and even so, when there are some artifacts, this approach can extract some information from slides without artifacts and make a prognostic signature. So now we are in further analysis together with Institut Gustave Roussy. We also found that we can identify a subgroup of HPV plus patients who doesn't seem to benefit in 2 independent data sets from chemotherapy. So radiotherapy seems to do as well as concomitant chemoradiotherapy. And we thought that could be perhaps an objective way to go for those de-escalation trials. So rather than de-escalate to all HPV plus, which is not totally satisfactory because finally the results are not perfect with HPV plus, to try to identify a favorable group with radiomics and then de-escalate specifically to this subgroup. So we believe that that's probably more relevant for you, that this approach, that's more sort of more virgin area, I would say, is also very interesting for window trials. And then I have to introduce the concept of 4D radiomics, where rather than doing imaging analysis just before the treatment, you do it repeatedly at different time points, and you look how certain features are evolving during time. And for example, in liver metastases treated with neoadjuvant chemotherapy and with surgery, we can actually predict very well the pathological treatment response and earlier and better than RECIST, because we can see some texture changes which are even not visible by the human eye. And there is also a recent trial that we just completed in lung cancer, where we did perfusion CT, hypoxia CT PET, and then we give the simple nitroglycerin patch used in cardiology during 48 hours, and we did a second perfusion and hypoxia scan, and in some, not all patients, we see, especially in the patient with a low blood volume, we see an improved perfusion and decreased hypoxia. So it's a typical window trial based on imaging. And this is, for example, one case where in yellow you see the hypoxic areas. You could see visually it decreased 2 days later. But the interesting thing is that if we remove the PET and analyze the CT, we can also see a change, a consistent change. And that's quite interesting, because in multicentric studies, these H64 PET scans are quite difficult to implement. So what about toxicity? We all know examples of great drugs which fail due to toxicity. So we would like to have a hand on it to try to eliminate the patient with high risk of toxicity, and when there is toxicity, try to quantify it. For example, for lung, it's actually easier than with tumors, because the segmentation is very straightforward, it's totally automated. And for tumor, it's semi-automated. So you can easily quantify some disease like COPDs with numbers. And now, just for reference, the gold standard is the 6-minute walking distance, which we'll agree is very functional, but is highly variable and not very easy to implement. And when there is a toxicity, you can also quantify it in different ways. So, in conclusion, I hope I convince you that it's possible to extract in a robust way more quantitative and clinically relevant information from standard imaging. And of course, the more you standardize the imaging, the better are the data. That's what we call radiomics approach. And we hypothesize that radiomic is correlated to biology. We have shown it for HPV and recently for hypoxia. Can improve accuracy of prediction, for example, non-small cell lung cancer, inoperable and non-metastatic. And that still has to be worked out in more detail, can help us to quantify the effect of treatment with 4D radiomics and the effect of treatment on tumors, but also on normal tissue. So in conclusion, I think our PACS is actually a gold mine of data. It's a mineable source of data. Some people would say, okay, if you compare to diffusion MR and etc., radiomic is the poor man biomarker. I don't have a problem with that. But I would say radiomics is a simple, quantitative, and robust biomarker ideal for drug development and multicentric study. So I would like to thank all my colleagues, especially Ralph Lejnar, Sean Walsh, and I thank you for your attention. So I'm also the chairman of this session, so any questions?
Ask you a question by Charlie. So thank you for your talk. Do you have any plans to prospectively validate this approach?
Yeah,
because
there are several trials running at the moment. If you go to clinicaltrial.gov, you will see, you type radiomics, you will see there are several trials running at the moment.
Thank you.
Yes?
That was a great talk. I was wondering if you've applied these techniques to immunotherapy-treated patients just yet?
Sorry, could you repeat the question?
If you've applied these techniques to immunotherapy-treated patients?
Yeah, that's an excellent question, of course. Because an obvious hypothesis now, we are aware of the great work showing that non-synonymous mutations translate to more higher presence of neoantigen and more immunosensitive tumors. An obvious hypothesis now is to see whether we can also quantify this non-synonymous mutation, presence of neoantigen with radiomics. So we are working on that, and several groups are working on that.